Welcome to this lecture on fatty acid and triacylglyceride biosynthesis. This lecture is based on chapter 21, and here are the pages that you should focus on in the Leninger on the 7th edition. Here, your goals are to learn how we synthesize fatty acids, focusing on palmitic acid first, and how we can assemble these fatty acids into triacylglycerol with glycerol as the backbone. So these reactions take place in different areas of eukaryotic cells. Today we'll focus on fatty acid synthesis, which takes place in the cytosol. And then once we can further modify this fatty acid by either elongate them, so adding extra carbons, or we can introduce double bonds. That's called desaturation of fatty acids, and that takes place in the endoplasmic reticulum, or some very long fatty acids in mitochondria. But I have also a lipid course called MOL200. If you're interested, we also look at some biochemical reactions, and in particular phospholipid synthesis, as well as cholesterol synthesis in the early and late stages. And that's covered by MOL210 later on. Now, where do these fatty acids comes from, and why do cells end up generating them actually? So this is a key question because, as you probably are aware from what you eat, most fatty acids are actually obtained from the diet, from what you eat. But in the case of excess carbohydrate intake, and when an organism has met the energy requirement, so you can actually store excess carbohydrate molecules or carbons into fat, so into triacyl glycerides, and we'll go into this into more details. And this happens in the liver, but also in adipose tissue, and sometimes in the mammary glands that are undergoing lactation. So this is the de novo. This is called the de novo synthesis. What we can make is palmitic acid with carbon 16, myristic acid carbon 14, and stearic acid 18 carbons. Right, so let's start with fatty acids. So this is actually a process of combining 8 molecules containing 2 carbons, and these are acetyl groups from acetyl coenzyme A, which will form here a 16 carbon saturated fatty acid, or palmitate, which is depicted here. And then palmitate can then be modified to give rise to other fatty acids. It can be elongated, giving rise to longer fatty acids, or it can be desaturated, giving unsaturated fatty acids. And as I said, fatty acid synthesis occurs in the cytoplasm. So where does this acetyl-CoA comes from? And as I said, this is exactly in the case of excess carbohydrate diet, which will be then in the form of glucose uptake. And when the energy requirement is already met, so this glucose needs to be stored somehow as fat. And that's because from glucose uptake, which is not depicted here, we follow glycolysis, and eventually we end up with these acetyl-CoA in the mitochondrium here. So this will not go through the citric acid to generate more ATP, because we have, in this case, there's enough ATP being generated. So what happens instead is that these acetyl-CoA will be fed into fatty acid synthesis metabolic processes. But in order to do that, because the biosynthesis occurs and the enzymes are found in the cytosol, these acetyl-CoA somehow needs to be fed and transported out of the mitochondrium into the cytosol. And this takes place not directly, but via the transport of citrate, because citrate transporters exist in the inner membrane of the mitochondrium. So acetyl-CoA will combine with oxaloacetate to form citrate, which will be transported out. Then once it's in the cytosol, citrate will be transformed into acetyl-CoA via the citrate lyase, therefore regenerating oxaloacetate. This then requires 2 molecules of ATP here, because we need the generation of oxaloacetate from pyruvate, which and another molecule of ATP here to generate acetyl-CoA from citrate. Right, and then these reactions in the cytosol, these biosynthetic reactions, requires NADP or NADH, and this then is being used in these reactions to return pyruvate or malate back into the mitochondrium, so that we can use new molecules of acetyl-CoA to feed into this process. So the first reaction, now that we have our acetyl-CoA in the right place in the cell, and these acetyl-CoA will be first used in the reaction to generate a 3-carbon, so from a 2-carbon to a 3-carbon intermediate called malonyl coenzyme A. And this is synthesized by the enzyme called acetyl-CoA carboxylase. Here is the reaction. Here's 1 molecule of acetyl-CoA, bicarbonate, ATP, and this adds an additional carbon, generating a 3-carbon molecule, so malonyl coenzyme A, and ADP plus inorganic phosphate. The enzyme reaction, the acetyl-CoA carboxylase reaction, takes place in 2 steps. This enzyme consists of 3 functional domains, and each have a different enzyme activity. You can see them depicted here in this diagram. First, a biotin carboxylase, a biotin carrier protein here with the lysine chain bound, conjugated to a biotin molecule, and a trans carboxylase. So first of all, the biotin carrier protein will receive the bicarbonate molecule, and this is ATP dependent, ATP dependent reaction. Then this will act as an intermediate to allow conjugation to this CO2 is then transferred onto acetyl-CoA to generate malonyl-CoA here. Finally, the last step is to the release of this molecule, which is now ready for our building as a building block for generating a long hydrocarbon chain of fatty acid. So as I said, malonyl-CoA will be our building block to generate a long chain of hydrocarbon fatty acid, and this is done by an extension by 2 carbon units, which is donated by the activated malonate, so malonyl coenzyme A. There's a loss of CO2 at each step, which is the CO2 that is gained in the ACC reactions to generate malonyl-CoA is lost as soon as this malonate is donated to this fatty acid synthase reaction. And as you can see, this is our malonate CoA, which is donated here, and this chain, the hydrocarbon chain, elongates. A new one comes in here, which will be then donated here. So you can see there's an elongation by 2 carbons at a time, and so on until we reach the full chain of palmitic acid of 16 carbon. So this is done by, as I said, type 1 fatty acid synthase, FAS. There's no intermediates that are released until the end synthesis of palmitate. So all of these intermediates are covalently linked to the enzyme. So this reaction is actually divided into 4 steps for each addition of 2 carbons. Here's our malonyl group here, which is bound to the enzyme via a thioester bond. An acetyl group is also conjugated to the enzyme as well, and this is the very first reaction that requires an acetyl group and a malonyl group. Thereafter, it will be only malonyl, which will be donated. So the first step is a condensation, which releases CO2 onto the enzyme between the acetyl and the malonyl. The second step is a reduction that uses NADPH, and finally a release of water via dehydration, generating this double bond here, and a final step of reduction. And now we have a nice saturated 4-carbon molecule still anchored onto the enzyme. Here is the crystal structure of a mammalian fatty acid synthase type 1. So most of it has been resolved, and you can see how big and complex this protein. So it's a multifunctional polypeptide. It is very large, of 240 kilodalton. And what is so interesting about it is that it contains 6 different catalytic domains. As depicted here, they are color-coded in the crystal structure here. So these each will be helpful in either conjugating malonyl CoA and the acetyl CoA onto the enzyme, conjugated covalently, and also each of these domains will carry out the 4 different steps that I've mentioned. But what I would like to point out is 2 specific domains here. Is this beta-keto-acyl ACP synthase, acyl carrier protein synthase. This is where the growing fatty acid chain is covalently attached. On here, you can see it sort of sticking out here. And this is the starting point where the acetyl group is donated. So 1 acetyl molecule. Whereas the malonyl group is conjugated here covalently to the acyl carrier protein, which consists, so this is part of the protein. There is a serine chain which is conjugated to vitamin B, and here on this SH group, this is where the malonyl CoA is attached. So these 2 molecules are covalently attached and don't leave unless, apart from the transfer of the malonyl CoA onto the acetyl CoA. Now, here are the exact steps happening. first loading of the acetyl group on the case domain here of the enzyme. We don't have to go into the details, but you can see how these diagrams are color-coded, follow the color code of these various domains in the linear version of it, and in the complex, in the crystal structure of the enzyme. So this is the loading of the acetyl group and the release of the coenzyme A. Here. Finally, loading of malonyl group onto this ACP here, so that these two molecules can now conjugate, and the CO2 can be donated onto the acetyl group. There's a release, so this is the condensation step. There's a release of CO2, and now it's ready for the next. It's for the next step. So these you don't have to learn by heart, but this is to explain what each domain is involved in. The reduction. This is the reduction here in step, targeting this beta carbon here, and it's catalyzed by via NADPH. So this reduction step. And now there is a dehydration step, releasing water, and finally the additional reduction to generate this time a 4-carbon acyl chain called butyryl ACP. ACP because it's still conjugated to the acyl carrier protein. Now, this butyryl group is then translocated from this end of the protein onto the acyl carrier protein. And this is the end of the first round to generate a saturated 4-carbon chain. Now, this means that this is free. We can load this time a malonyl-CoA. So this is we don't use acetyl-CoA. We use a malonyl-CoA, which is made by ACC, as I explained earlier. And then the same process occurs, whereby these 2 carbons are donated to the butyryl group. And so on, and so on, and so on, until we until we generate a full palmitic acid. So this is the step-by-step synthesis of this fatty acid here that are summarized in this figure. So as I said, don't remember every single domain that is involved in this in the enzyme reaction. But the important thing is the fact that we use 1 acetyl-CoA, 1 malonyl-CoA to generate a 4-carbon butyryl ACP, and this is the very first steps to finally generate a long 16-carbon fatty acid chain. So overall, in sum, we need 7 cycles for the synthesis of a saturated 16-carbon fatty acid chain, and to release a free palmitate. 7 malonyl-CoA are needed, and these needs to be synthesized by ACP at the cost of 7 ATP. Yes, so that makes our 14 carbon, and we also need 1 acetyl-CoA, 1 free acetyl-CoA, which is not transformed to malonyl-CoA. So there are 7 cycles that consume 14 NADPH, and in sum, altogether, we need 8 acetyl-CoA, so 1 acetyl and 7 of them that are transformed into malonyl-CoA, 7 ATP, 14 NADPH, to generate 1 16-carbon palmitate and the release of 8 coenzyme A 7 ADP and 14 NADP plus 6 water molecules, not 7, because one molecule is consumed during the release of palmitate at the end. So, yes. As mentioned earlier, so these reactions takes place in the cytosol, and this is to allow the segregation between anabolic and catabolic reactions that we'll go through next. So the anabolic reactions, synthesizing fatty acids, takes place in the cytosol. This is in the same compartment where we have NADPH available for the reductions, there's 2 reduction steps in the synthesis of this hydrocarbon chain. So this is in the cytosol in animal and yeast cells, so eukaryotes of animal or yeast origin, but in plant cells it's found in the chloroplast. Synthesis of fatty acid needs to be tightly regulated, and this is the case when there is enough citrate in the mitochondria, and this citrate then will act at high concentration, will act as an allosteric activator of the ACC, the acetyl-CoA carboxylase. So this reaction converting acetyl-CoA to malonyl-CoA, a 3-carbon intermediate, is a rate-limiting reaction. So therefore regulated by citrate. In addition, the end product, palmitoyl-CoA, will have a negative feedback on this reaction. In addition, in terms of negative regulation of fatty acid synthesis, is the hormonal dependent regulation, which leads to downstream signaling, which eventually will modify the protein. It will phosphorylate the protein and inactivate it. So these are ligands that will act via G protein coupled receptors, for example. So they will trigger a cascade leading to the phosphorylation and the inactivation of the enzyme. What about the fate of palmitate? So we have made our 16-carbon long chain, hydrocarbon chain, and this can be further desaturated into palmitoluate. So there's one double bond introduced at carbon 9, between carbon 9 and 10, or it can be further elongated by the addition of 2 additional carbons, which leads to the generation of stearate. This then can also be further elongated into longer, fully saturated fatty acids. Or alternatively, this can also be desaturated, again at the same carbon, so desaturation at carbon 9 to oleate. Now, so these steps, these elongation and desaturation, can occur in the smooth endoplasmic reticulum in the mitochondria. Elongation uses malonyl-CoA, just like we have done, to elongate the chain to reach palmitate. Now, desaturation on this carbon between carbon 9 and carbon 10 in mammals is possible, but we can't desaturate beyond carbon 10. We don't have the enzymes that will recognize, we don't have the enzymes that desaturates beyond this carbon. But plants do, so then we are dependent on generating polyunsaturated fatty acids due to our diet and ingestion of plant various plants. So, and this leads to the generation of these polyunsaturated omega-6 or omega-3 from this first desaturation in plant on carbon-12, which leads to linoleate. Then there is some further desaturation to generate ALA, so alpha linolenate, or gamma linolenate. And you can see that these can feed in further through elongations and further desaturations only in plants to generate these very polyunsaturated. There's 5 double bonds in EPA here, and here 4 double bonds for arachidonic acid. And these polyunsaturated fatty acids of the omega-3 and omega-6 are important also in various processes, including inflammations. So they are further metabolized into other products involved in chronic or acute inflammation. So the fatty acids that are either synthesized due to excess carbohydrates or ingested from the diet, they, depending on therefore on the physiological status, they are converted to triacylglycerol for storage of metabolic energy. Or they can also be used to generate a membrane in the created synthesis of phospholipids. And for this, glycerol 3-phosphate is required. This is the backbone that will either be used to generate triacylglycerides or phospholipids here. So you see 3 fatty acids in 3 acylglycerides, or 2 fatty acids and a head group in membrane phospholipids. So where does this glycerol 3-phosphate comes from? So it can either come from the product, one of the byproduct of glycolysis, which is dihydroxyacetone phosphate, which is reduced to glycerol 3-phosphate. Or it can be due to the phosphorylation of glycerol by glycerol kinase, so an addition of phosphate here on carbon-3. So not all tissue can can phosphorylate glycerol, but the liver and kidney can do that, not adipocytes. So adipocytes will get their glycerol 3-phosphate from glycolysis and from dihydroxyacetone phosphate. So now that we have our glycerol 3-phosphate, we can now add 2 fatty acids on this molecule. And these fatty acids here are represented with an R1. R represent the hydrocarbon chains of a certain number of carbons. So these fatty acids are first activated as fatty acid CoA by various acyl-CoA synthesis, which will recognize different types of fatty acids here of different chain length. Often this first fatty acid is an saturated fatty acid, and as I said, there are different forms of these acyl-CoA syntheses recognizing different chain lengths of fatty acid. So this fatty acid, which is activated, is then transferred onto this carbon 1 of the glycerol 3-phosphate backbone by an acyl transferase. Again, there are many different forms of these enzymes. This will generate a lysophosphatidic acid. Lyso because it's a common terminology to indicate that there's only one fatty acid that is present in the molecule. So this lysophosphatidic acid will now receive a second fatty acid activated in the same way. Often this is an unsaturated fatty acid, and again there'll be different forms of these acyl-CoA syntheses and acyl transferases as well, using this as a substrate. So now we have these particular molecules with 2 fatty acids, 1 phosphate here in carbon 3, and this is called a phosphatidic acid. a phosphatidic acid, it is a glycerol phospholipid. And this is the simplest of the glycerophospholipids. Now, this phosphatidic acid can be used as a precursor to generate both triacylglycerol or glycerophospholipid, important component of membranes. But if we focus on the left here, to generate triacylglycerol, our phosphatidic acid needs to be converted to diacylglycerol, which by a phosphatidic phosphatase. So this phosphatase will remove the phosphate group here to free up this hydroxyl group to be able to transfer a third activated fatty acid here onto the molecules, hence generating triacylglycerol. So this part, synthesis of glycerophospholipid, important for membrane, is covered by MOL210. So this so triacylglycerol synthesis is highly regulated as well, and this is due to the action of insulin. So insulin will convert these excess dietary carbohydrates or excess proteins to generate acetyl-CoA, and insulin has a positive effect on the generation of acetyl-CoA and further the elongation to generate fatty acids. So triacylglycerol is taken up eventually and stored in the adipose tissue, so in the cells that committed to store TAG in these lipid droplets. And when TAG is needed, well, it can be metabolized and mobilized to fatty acid can be released from this molecule, and fatty acid can then be transported to other tissues such as the muscle for energy. And the remaining fatty acid is then taken up by the liver and reconverted to triacylglycerol with the use of glycerol 3-phosphate. Then this TAG is then resent to the blood circulation and taken up by the adipose tissue. This is called the triacylglycerol cycle. Now, so in this chapter, we have learned that the fatty acid synthesis is a multi-step process. It starts from acetyl-CoA that needs to be released to the cytosol, and it's converted to a carboxylated product, malonyl-CoA. Malonyl-CoA is then the building block to generate long 16-carbon chain. In fact, these fatty acids are then converted to a particular phospholipid called phosphatidic acid, which is the precursor to generate triacylglycerol. So triacylglycerides are built on a glycerol backbone that is derived from glycolysis. So this product of glycolysis, which is dihydroxyacetone phosphate, or glycerol, which is then phosphorylated. Yes, and then we haven't covered the cholesterol aspects, but in an incorrect traffic of TAG is actually correlated to multiple human diseases. And so, for example, excess carbohydrates, which is stored into TAGs, can have detrimental effects at some level and can contribute to the metabolic syndrome.